welcome back. Today we will discuss the second unit of microbiology. The name is General Characteristics of Microbes. In earlier video we discussed about the introduction of microbiology, in which we studied what are the branches of microbiology, the definition of microbiology along with their historical aspect, then significance and relevance to the nursing and scope of microbiology in nursing. In this unit we are going to understand the following things. See the lecture outline. First we will discuss in this particular lecture the structure and classification of microbes. Then we will study the micromorphological types, then size and form of bacteria, motility, colonization, growth and nutrition of microbes, temperature, moisture, body and their blood and their fluids, body fluids. Then we will discuss the types of staining, then culture and media preparation, and then laboratory method for identification of microorganisms. In this lecture we will discuss each and every microbes under the following things. If microbes emit or they show the pathogenic activity, they are known as pathogens. So we will discuss each pathogenic microbes under these following topics. First one is classifications, where those pathogens will occur, then their localizations, okay, then their morphology and culturing. Morphology means their out structures, and their culturing, how we can cultivate those pathogens. Then we will discuss their developmental cycle, then pathogenesis and clinical picture, then diagnosis, therapy, and epidemiology and prophylaxis. Let me tell you the bit about these terms. Classification, as you know, it's just a division of microbes, okay, so that we can identify those microbes in particular groups, okay. Then localization. Localization means basically the habitat where these microbes can be found. Then we will study the morphology, the structure. Morphology is basically a term used for the external appearance, how one is looked, how one's bacteria is looks externally, that's called the morphology. And culturing. Culturing is just a cultivating whatever pathogens is found inside the human body or other animals body. We can take the fluids, we can take the appropriate fluids, and then we put into the culture plate, the media plate, and we can grow. This phenomenon is called the culturing, okay. There are so many types of culturing we will discuss in our successive lectures. Then developmental cycle. Developmental cycle means how one pathogens grow, multiply, and enhance their progeny, increment their progeny, increase their progeny. That's called the developmental cycle. Developmental cycle is very much necessary to understand, because there are some diseases, we can say the malaria, okay. In the case of malaria, there is one proper cycle, okay, and there are some anti-malaria drugs which disrupt, which inhibit the developmental cycle of malaria, okay. Then pathogenesis and clinical picture. Pathogenesis means disease development, how one pathogen enters in body, they destroy the immune systems, which is a protective system in our body, okay, and then how they colonize there, they increase their number, they capture our cellular machinery, okay. So all those sort of things we will discuss in the pathogenesis, the clinical picture. Now if we are infected by one microbes, one pathogens, then there are some altered body appearance. So sometimes we have rashes, sometimes our homeostasis mechanism is altered, like our temperatures got high, body shows the inflammation process, then we have the redness of area, then skin lesions. All these things is called the clinical picture, basically the sign and symptoms, okay. Then diagnosis. Diagnosis is to making the decision that a particular person is suffering from a particular disease. That's called diagnosis. In diagnosis, both laboratory technique, imaging techniques, all are implied, all are used. Okay, then therapy. After diagnosis, we will plan a proper treatment, okay, a proper care by which a patient can recover. So for that, there are the therapies. We can say the antibiotic therapies, we can say some non-invasive therapies, invasive therapies. Okay, sometimes we have to perform the surgery also to discard the microbes, to discard the pathogens. Then epidermology and prophylaxis. Epidermology means there are, you know, like in our whole like planet, some areas are known for some particular pathogens. Other area is known for other kind of we can say the pathogens. Every pathogens cannot grow in every environment. Even though bacteria is mastered, the pathogens, some of the pathogens is mastered in the art of adaptations. They can adapt so many harsh conditions, as I already told in the previous lecture. They can tolerate as high as 1000 degree temperature. They can tolerate the ultra acidic environment. They may be found in the deep in the oceans. So all those things they can, but every pathogens cannot. Okay, so it's better to it's necessary to understand the epidermology of a particular bacteria, so that one can aware, okay, one can aware that what kind of pathogens is present in their area and what they can cause the symptoms. Clear? Now, prophylaxis. Prophylaxis basically we can say like prophylaxis is basically one infection, okay, which can occur by the pathogens. Okay, prophylaxis. In these whole, if I take one bacteria, for example, Staphylococcus, if we take, then we have to classify it like gram positive, gram negative, aerobic or non-aerobic, their nutritions, then their localizations, then morphology, how the Staphylococcus is appearing, then their developmental cycle, like all the reproduction, growth, multiplication, all those processes. Then pathogenesis, how Staphylococcus enters in our body, which part they affect. Okay, then their clinical picture, their sign and symptom, what kind of sign and symptoms is caused by the Staphylococcus, then their diagnosis, which stain, which culture is necessary to diagnose a particular this Staphylococcus bacteria, then their therapy, which kind of antibiotic is applicable to this, and where this bacteria is found, like tremendously, or we can say in a large number. And then prophylaxis, if we not manage this bacteria, then what kind of reactions our body shows. Okay, so we will discuss each and every pathogens under these topics. Hope you all have the clear with this concept. Now, structure and classification of microbes. As you know, in your previous classes, you studied about the 5 kingdom classification given by exactly the R.H. Whitaker. R.H. Whitaker is one, we can say, the taxonomist, okay, who divided the whole organism into the 5 category. Okay, into the 5 category. They chosen the criteria by which they classified. The first criteria is the cell wall, okay, structure of cell wall, then the mode of nutrition, okay, then they take the type of reproduction. These 3 are the main criteria in which, in this basis, R.H. Whitaker classified the whole organism present on the earth into the 5 category. The first category, as you saw in the slide, is Monera. Monera is basically the kingdom of prokaryotic organism, okay, prokaryotic organism, and primarily in this category bacteria is present. So bacteria is the sole member of Monera. Clear? Now come to the Protista. Protista comprises the unicellular eukaryotic organism. For example, protozoa and algae. Protozoa and algae are come under the kingdom of Protista. Clear? Now, fungi. Fungi, as you know, it's also one of the most causative agent in our body. It also causes so many deadly disease. Okay, and as well as we have so many beneficial point of view related with the fungi. Okay, so in fungi they include the multicellular eukaryotic organisms like mold, yeast and mushrooms. Then plantae. For plantae consists of the multicellular photosynthetic organism like the plants. So there are few plants. Why I am putting this plant, because we are studying the microbiology. And what is the use of the plants? Okay, there are some allergic reactions called the hay fever. Okay, hay fever is due to the inhalation of some of the pollen grains. Okay, there are some species of the plants, their pollen grains can affect our respiratory tract and they can produce the allergic reaction basically. Okay, so allergy is some of the plants, I will discuss it in later. Then there are some plants which can cause the allergic reaction to the human body. Then animalia. Animalia encompasses the multicellular heterotrophic organism referring to the animals. Now I have one question, where we are classified among all these 5 kingdoms, where we are classified. So comment down where we are present in this 5 kingdom classification. Now there are some points related with the 5 kingdom classification. The first point is, in the 5 kingdom classifications all organisms are classified except 2. Can you name them? Yes, first one is virus and second one is prions. See, why RS Whittaker not classified all these things? Because they chosen some of the criteria. In previously I told you that they chosen 2 criteria. The first one is the cell structure, cell wall structure, then mode of reproduction, and then the phylogenetic relationship, like evolutionary relationship. Okay, so these all things is not found in the virus. Virus is like the borderline between the living and non-living. It's like in the borderline. They have only the nucleic material, we can say the genetic material, but they didn't carry any type of cell organelles like the mitochondria, Golgi body, ribosome, okay, centrioles, nothing. They don't carry anything. Even though they don't have any kind of like cell wall, they don't have the cytoplasm. So RS Whittaker thought they don't fulfill their criteria, then how they can classify these viruses? Same apply to the prions. Prions are basically like sometime what happened due to some mutations, due to some alterations come from the environment, external environment, our own proteins become pathogenic. Okay, they can disrupt our normal body physiological function. So that's proteins, misfold protein is called prions. So prions are also not categorized in the 5 kingdom classification. So these, we can say these 2 are the drawback. Okay, so okay, let's read these paragraphs. The 5 kingdom classification system proposed by Robert H. Whittaker was comprehensive, but there are certain organisms that not fit neatly into this scheme. Okay, one example is virus. Virus don't exhibit the characteristics of living organisms outside a host cell and are essentially genetic material, either DNA or RNA, surrounded by a protein coat. As such, they are not classified within the 5 kingdoms because they lack cellular structure and independent metabolic processes. Okay, once virus enters in our body, they hijack our own cellular machinery. So they don't have their own, they are not independently functioning, they are dependent on the living entities. Clear? Then other entities like the prions, which are misfolded proteins associated with certain disease, also do not fit well into the traditional classification system as they are not complete self-sustaining organism. Okay, now what happened? After some time, one scientist come, one evolutionary, one biologist come. His name is Carl Woese. Her name is Carl Woese. They also classified the organism into the domains. Okay, into the domains. So the first domains he classified is bacteria. In this category, only prokaryotic cells containing organisms are classified, and already we know prokaryotes is bacteria. So the primitive bacteria is classified in next category, that's archaea, but the normal bacteria is classified into the bacteria domains. Then come to the archaea. This archaea contains the other kind of primitive bacteria, we can say the old bacteria, which can tolerate the extreme environments, okay, which can tolerate the extreme environments like the thermoacidophils, okay, which can tolerate as high as 1000 degree temperature. They may be present in deep inside the ocean, deep inside the earth, okay. So all those primitive bacteria is classified under the domain of archaea. Then eukarya. In the eukarya, all the eukaryotic organisms ranging from the unicellular microbes to the complex organisms like us, they all are classified into the eukarya. So if I take the classification criteria, first is 5 kingdom classification given by the R.H. Whitaker: Monera, Protista, Fungi, Plantae and Animalia. Drawback 2, first is virus, second is prions, okay. Then come to the Karlsruhe. Karlsruhe classified all those organisms into the 3 things, first 3 domains basically. The first one is archaea, first one is bacteria, second one is archaea, and third one is animalia. Am I right? No, eukarya. Clear? Okay. Now see the beautiful diagram. As you see, I zoom the slide. That's a HIV virus structure. See, you see these normal spikes, see these normal spikes, these normal spikes are basically the infected part. These are basically the infected part, and they is meant for adhering on our cells, okay. Basically they destroy in us the lymphocytes, type of T lymphocyte, B lymphocyte. They attack, HIV virus mainly attack the T lymphocyte and B lymphocytes. And you see the bit envelopes, crisscross envelopes. All these envelopes is just a covering of their genetic material. And now see the Escherichia coli. This Escherichia coli, they are naturally present in our body. Sometimes we acquire, and it's the most responsible for urinary tract infection, okay. See the structure, these all are the microscopic structure. Now the protista, example euglenoids. This is a protist structure. I will discuss all these structures in their particular topic like bacteriology, virology, mycology. Now this is the fungi. See how beautiful it is. Okay. Now what we do, we will take each category of microbes and then study in a detailed way. So today we studied the bacteria. So for bacteria, study of bacteria is known as bacteriology. It's a big branch, it's a broad branch. Under we will study the bacterial morphology, their appearance, their external appearance, then their normal physiology. I mean, like bacteria is also a living organism, no? So they have their own mode of functioning. Their cellular mechanism is functioning on a particular way. Then every living organism, including viruses, contain their genetic material, because the genetic material is the one which is responsible for transmission of character and continuity of the generation. That's the genetic materials. So always keep in your mind that genetic material is something which is present in every living things, okay. There is no any exception. Clear? Then ecology is basically the habitat. We will also discuss the bacterial habitat, that is a particular kind of a specific bacteria is present on the specific kind of environment, because I already told, no, every bacteria cannot tolerate the every temperature, even though they have the appropriate enzymes by which they can tolerate the unfavorable situation, but they cannot tolerate in a longer way, they cannot in a longer period, they cannot tolerate. Okay, sometimes comes their population will discard it from this their non-native places. Okay, then their pathogenicity. Pathogens means disease causing ability. Okay, so under the bacteriology we will discuss the morphology, physiology, genetics, ecology and pathogenicity. Hope you all are clear with these terms.